Cool. Thank you very much for the invitation. Let's see. So I would like to discuss today with you health, actually, not really disease. And this is a short session on AI, or title in AI. So and we think a lot about what does it really mean. Also, Gadi tried to give you a glossary. But I would argue that many people think about it very, very differently. And in a way, people that are coming now from this big revolution of large language model and the ability of those machines actually to imitate human thinking is, in a way, amazing and encouraging. But on the other hand, us as biologists must somehow draw the line and ask ourselves, what is this doing actually, even if it is becoming superhuman in its, let's say, deduction capabilities? And so a superhuman AI may be like better than any of us as a physician or as a human thinker, but it doesn't really mean that it's going to solve biology, because biology have in its like essence no relation to human thinking, except maybe for the fact that it is implementing human thinking. But other than that, biology is working for itself. And we think that, I think that it's going to be amazingly interesting to rethink what we as humans used to define as health and disease, which was inherently shaped by our ability to think and to classify and to process data and to, in the day-to-day practice of a human physician, make decisions. So perhaps looking into new biological and computational ways to define what we perceive as pathophysiology, rethink it, is going to be maybe our biggest challenge. And I'll give you just very brief examples of what we are trying to do in these directions in practice, because these are big words and you have to, by the end of the day, come with actual data. So the first example, which is going to be very brief, is published a while ago, is to ask ourselves, as humans, we tend to define normal and abnormal health and disease. The first thing we are usually doing, or physicians are doing, I'm not a physician, is to take a lab test, to take a piece of data and declare it normal or abnormal. So we had the privilege of trying to revisit those normal ranges or those ideas of what is going to be normal if you just come to a physician. And for example, we are looking at your complete blood count. So together with Gabi Barabash, we thought a lot about those problems in the context of population health. Neta and Aviezer in the group, we had the privilege of looking at like a very, very large scale data set of over 4 million Israelis with over 20 years of history or complete history, where most of this history pertained to life phases of those individuals where they were healthy. So the first thing we could do was to look at their lab tests, just their lab readouts. What you see here is statistics that you cannot probably really read it, but just look at the colors. It's just trying to assess how much of the data we are accumulating in electronic health records actually are defining what healthy people, or people that we define today as healthy, are doing. So it turns out that a lot of it, the x-axis here are always the age. And you see the numbers, so how much data you are collecting for people that are defined currently as healthy or are not healthy. And again, we have not only the lab records of those individuals, but all their diagnosis and other things, so we can classify at least what the system is thinking about their current position. So the first thing you'll immediately know that if you just look at so these are just numbers, so how much data we have, but here are the values, the distributions over age of different types of lab tests. And of course, all of them are changing with the age. All of them have huge variance inside the population. And what we define as abnormal, like out of the range, is quite a poor definition of what we may be thinking to do with this data. So for example, Here are age-linked progressive anemia, which is something that you'll always see, all kinds of transient increase in liver enzymes that you may not have think about. Of course, in women, perimenopause effects. So all of this can be modeled statistically, like in a different way than just saying it's normal or abnormal. This is not AI, this is statistics, okay? But it is very useful to have. And the thing that you immediately observe when you look at these types of data is that it's not only that one definition of what is normal and abnormal, let's say in your neutrophil count, is let's say imperfect, or you can do much better. It's also can be you can do much better with personalizing this estimation because of germline genetics or other things that happens to a certain individual. You can define and observe autocorrelation or a lot of persistent behavior of individuals when you repeatedly sample them over the years. So some of them just tend to have high neutrophil counts, of course. Some of them have lower, and the normal of the individual with, let's say, germline high neutrophil counts is not the normal of the other guy that have the low one. So all of this is something you can pull out from the data and plug into any model you can imagine. So this is published. You can look at the paper and maybe use the models. But we can then dive into this data further and ask the same question about diseases. So according to, let's say, the EHR, or according to the medical textbook, okay, there are tables that defines when we are crossing a certain threshold and become sick. So we get an ICD-9 or ICD-10 diagnosis code. And then someone will do something with us, hopefully. But it's not, probably biology is not working like that. It's a process. It's a spectrum of effects that are continuously evolving. And it's very, very interesting to think about them or to try to model them this way. So instead of this dichotomy of health or disease, this is also published work more recently, went into the data and looked, for example, at the most prevalent chronic diseases that are emerging with the ages. Okay, you see those diabetes, liver, kidney, of course cardiovascular, and all of them are, of course, increasing in the incidence and prevalence with the age. So this is kind of a process that seems almost unavoidable, but you can now look at the trajectories of those individuals that are going to develop these diseases and ask, when they were not classified as already having the disease, what was the distribution? How were they traveling in the health space toward getting this disease? I'm saving you a lot of details here because, again, we don't have time. Just mention briefly that you need some fancy modeling in order to achieve this nowadays. Because even with 20 years of history, you cannot take an individual at the age of 20 and look at his or her future at the age of 80. So you need to somehow infer from shorter-term histories, longer-term histories, and we have did it using some nice forward-backward approach where we infer a Markov model trying to reason on how individuals are going to progress from a health state at age X to a health state at age X plus 5. And then we can plug it into one model, and this turned out to be very nice. It can work, given that you have a lot of data on individuals at all health ages, and of course many confounding factors then are going to be associated with that. Some of them were mentioned today, people that were born in the 30s of the last century and people that were born in the 90s of the last century. They are not experiencing the same environment, nutrition, everything. But it's a good start. So using this type of modeling, you can now come up with a probabilistic estimation of what is the chance of a certain individual, given that it is he or she is now completely healthy, to develop any of the chronic disease I showed you before. And also you can try to predict what is the chance of this individual to survive, let's say, the age of 85. Look at the paper if you want more details. Just one crux here. The crux is that you can now classify individuals at any age, and let's assume now they are healthy. They are not in the disease cloud, they are in the health cloud, okay? Only based on their current EHR, mostly standard lab records, okay? you can classify their scores, here, those are the blue circles here, to develop each of the prevalent chronic diseases. Okay, so blue is good, red is bad. Okay, and you have a different score for each of them. And some of them are completely obvious, like you have horrible BMI, horrible cholesterol, and so you're going to be diabetic, okay? With high probability, so you're going to be red. It's no surprise, but you can quantify it very precisely. And then the outer circle defines your, let's say, longevity potential. So what is your chance to survive the age of 85? And there is a nice story behind it that we are not going to into it. Then you can classify those individuals in this health cloud and ask, what is the combinatorial risk of developing any of the chronic disease? What is the chance of getting in good longevity? And of course, people that are doing bad with respect to all chronic disease are also going to do bad. That's not a surprise. The nicer thing is that you find individuals that are doing perfectly well with respect to all of the chronic diseases we know about, but are going to do excellent in terms of longevity, and people that are not going to do so well. And these are probabilistic estimations. Maybe you don't believe it, okay? One nice test, you can wait now 8 years and see what happens, or 50 or 20. But you can try and go to the parents of those individuals and ask how well did they survive. By the way, this analysis that was done on Israeli populations, you can take the exact same scores, the exact same statistical expressions, apply them to the UK Biobank or to the NHANES data, and you get the same results. That's quite remarkable, without even doing too much renormalizations. So here are the parents of those individuals in the super healthy and just people that are making their best effort but are not going to be super longevity. And you see that there is a change in the life expectancy of those parents, which is over, let's say, 1 year or so. Again, based on data that is very partial and based on the assumption that the parents are going to do what the children are doing, which is, of course, imprecise. So this is just showing you that within the health spectrum there is much more than what we are currently modeling. And of course, just saying it is not so interesting in itself. We want to understand what are the mechanisms, or what kind of diseases or new ways to define a disease are being hidden there. So I don't know the answers. We can do some analysis, however, once we have data that is referring to actual biological processes that are that can be maybe implicated in conveying these positive or negative effects. So here are indicators that are definitely not doing anything, like glucose or BMI. The curves you see here represent the distribution or the median behavior for individuals in 3 groups of people that are currently healthy, okay? And some of them have excellent longevity potential and some of them mid or lower. All of them are being excluded completely from people that are at risk to the classical chronic diseases, okay? Which are a very large fraction of the population, but we are ignoring them for the moment because for them, at least we have a reason. We know what mechanisms are going to make harm, to do harm for them. But so we do not expect those classical, let's say, diabetes, CKD indicators to distinguish those individuals. There are some interesting things with the BMI, maybe not really important for now. But here are metrics which are more unexpected that are distinguishing those super healthy individuals from the plain healthy one. So it gives us some ideas on what we need to follow up. For example, alkaline phosphatase, we really don't know why this is distinguishing those individuals so well. All kinds of things that probably relate to metabolism and ability to remain, or at least to reject frailty at later ages. But here is another type of indicator that we see that distinguish those populations that is going to connect to what we are going to talk about next, which are all kinds of hematopoietic indicators. For example, the ratio between neutrophils and lymphocyte. This is something that is characterizing those people that are super healthy. So lower neutrophils, more lymphocytes, that's good for you. It may relate to chronic inflammation. It may relate to all kinds of things we don't understand or don't understand very well. But now the question is, can we understand the dynamics that drive, let's say, those hematopoietic differences between those individuals over many years of life? So this is just a summary of 2 papers and a lot of work that has been done in the group using a quite unique resource, but you really want to push it further into something that you can also understand at the molecular level and maybe intervene with. So now I'm going to describe you a work that we are doing together with Liran Shlush at the Weizmann. It's a fantastic friend and collaborator. And here are some people that were involved in the paper that was recently published, and I'm going to show you some results from this paper and then some unpublished results continuing it. Oren from my group, Nimrod from my group, also from Tel Aviv University, and Nili, an MD PhD in Liran's group. So what we are doing here is to try and see if beyond those EHRs and those complete blood count that are giving us something but are leaving much to be desired in terms of their expressive capabilities to tell us what is really going on within patients, maybe we can use modern biology, modern genomics, modern single-cell genomics to acquire more information about those individuals. So here's the pipeline we are running here. It is based on the ability to extract circulating hematopoietic stem and progenitor cells from individuals, which turns out to be very, very feasible. These cells are rare, but you can reasonably acquire them from individuals using, let's say, 20 ml or so. And then you can run them through modern pipelines, giving you single-cell RNA-seq by the end of the day and all kinds of other things. I'll spare you a lot of the technical details, okay? Let's just try to make some points. The first point is that you want to run single-cell RNA-seq on many individuals, and we did it by now on 400 or so individuals. But is it going to be robust? Like, one of the beautiful things about genomics that all of us really, really appreciate is that it is, once you get an A somewhere, it's an A, okay? So it is super robust and reproducible. You'll go to the same individual twice, you get the same germline with noise you understand very well. Things are quite different, so they say, with single-cell RNA sequencing or with those techniques that are trying to understand a population of cell which is changing and are sensitive to all kinds of effects and biases. But turns out that this technology has matured remarkably, okay, if you know what you're doing, and if you're careful with your computation, then this type of manifold that you see here, each point here represents a cellular state that you characterize from the transcriptional state of a single cell or many single cells. And you see here the nice hematopoietic differentiation from hematopoietic stem cells toward all kinds of myeloid lineages, mostly erythrocyte or basophils, mast cells, and lymphocytes. So it turns out this is extremely robust. For example, when we are capturing this data from something like 200 individuals, okay, each of those states is being contributed by typically many, many dozens, 100 patients. And each patient will be mapped, the cells of each patient are going to be mapped very precisely and very quantitatively to this manifold. We can discuss later how and why this is sometimes not happening, but if you model it correctly, it can be done. So now you have something that is not just like a pretty picture, but it's actually classifying cells or providing you a new classification hierarchy for cells in the blood that are very close to the functions you may care about, which is how hematopoiesis is working at a certain individual. So what you see here are classes that are all coming from this manifold. So we take a couple thousand cells from individuals, project them on this model, and now we count how many of these individual cells are mapping into different hematopoietic stem and progenitor cells population. Just like we do with complete blood count, only now we are very sensitive and very much focused on a specific type of a specific type of cells. Good. So this gives us those distributions now of cells that are very progenitor-like, or lymphocyte progenitors here, or basophil progenitors here. And the ranges you see here are now quantitatively described how normal individuals, people that we considered as healthy, are distributed, just like complete blood count, only with different types of data. But the nice thing about it is that now that once you characterize a certain cell population from an individual, you have the entire transcriptional state of these cells. And if these individuals have some aberration or some misregulation, you'll be able to detect it. So here is data from many normal individuals, and the color coding, each bar is an individual, and the color codes describe those different cell states. The blues are those lymphocyte progenitors, the red are erythrocyte progenitors, and the yellow guys are those that are really MPPs, multipotent progenitors, almost stem cells, but not really. And so given a new individual, you can just compute how normal or abnormal this individual is. This is becoming very routine. And again, the premise is that it is robust, it is quantitatively reproducible. That's not trivial, okay? So now you can go and ask about questions about the emergence of disease. So let's think about the anemic phenotype, okay? Someone is developing anemia, okay? And as I showed you in one of the first slides, it's an age-related effect that is unavoidable, okay? All of us are developing slow anemia, okay? We don't consider it as a disease, but it is part of physiology. It's not good for us. But some people are going to deteriorate toward very rapid anemia. Eventually, some of them will become myelodysplastic syndrome patients. Some will deteriorate further toward AML. This is currently being diagnosed using bone marrow biopsy once people are having sustained anemia. So we want to change it now using those new blood samples. So bottom line, we can, okay, just doing profiling of circulating hematopoietic stem and progenitor cells from individual blood and doing a lot of things that are now published, okay, we can classify those patients very, very well. We think much better, actually, or better than what is the best practice at the moment, which is bone marrow aspiration and pathology. So here are just the AUC curve of this classification of MDS from just cytopenic patients. We now have to follow it up, of course, on much more individuals, but we are doing that. But that's not enough. Going back to the maybe first slide, we don't just want to take our current disease definitions, it can be MDS, and just do it better, although it's of course a very, very important thing to do, okay? We want to understand better the disease and maybe subclassify it. And here you see patients that are cytopenic, MDS, some of them are MDS or MPN, and you see their distributions, and actually immediately you see that there is more to it in this disease than what we originally thought, okay? There are individuals that are almost normal. There are subclasses with a lot of lymphocyte progenitors or almost none. Some of them are developing like a population of granulocyte progenitors inside the circulating. That's a new disease classification. We need to work on it. We need to understand what is happening. So this is part of the promise and the great excitement with those technologies. They come with a lot of data, with a lot of need in doing computational analysis that is, let's say, matching the degree of complexity of the data, but then hopefully we can discover new things, not just do better what we already do. So now we are following it up to the other branch of deteriorating hematopoiesis, in that case too many red blood cells, okay? So this can generate mostly polycythemia vera, and in some cases deteriorate into myelodysplastic myelofibrotic patients, myelofibrosis, and ET, where you have too many platelets or megakaryocytes. Here are similar data that from circulating hematopoietic stem cells of individuals that are in this spectrum. Again, polycythemia vera, ET, and MF. It turned out also in this case, it's very, very easy to take this data and just classify and maybe save the bone marrow aspiration. Just fresh statistics on how well we are doing, unpublished. And in that case, it's very easy also to see that just the current practice of taking complete blood count and the allele frequencies of the mutation that is driving a lot of those effects, which is JAK2, that's not enough. Okay, it's, on the other hand, single-cell RNA-seq is enough. So all of this is very nice, but again, for us as biologists, it is probably not enough if we want to understand what we are seeing. So we can take individuals that are healthy and classify them as high risk for developing a disease, but what are the mechanisms? Why are JAK2 or in polycythemia vera and MF, or why are the DNMTs and TET2 mutations that are generating clonal hematopoiesis that will later deteriorate into MDS? Why are these, or how is this contributing to the disease? We still do not understand. And I'm just previewing approaches that we are doing in the lab that can help us doing it. For example, isolating those circulating cells from individuals, tagging them with a clonal tag, and growing them in vitro under the influence of various cytokines turn out to be very, very feasible. And we are doing it now in the lab a lot, okay, with Liran's group. And this can tell us how the fact that you are mutated change your predisposition to differentiate it here or there. It's not the exact physiological conditions with all the hormonal signaling and all the cytokinic signaling that is happening inside an individual, but it tells you a lot about the potential of the individual clone to do what it is doing. Here are just very brief results showing you that this is still clones from normal individuals, how different medias are going to diversify the outcomes of those clones, and how you can get pairing between clonal fates from the same individual. If you want to talk about it, we can talk about it afterwards. But just thinking about the possibility of taking individuals that are predisposed to a certain disease and taking them to this assay is super exciting. Okay, so that's probably the end. The argument is, if we want to do more and we want to fulfill this promise of bringing what we have learned in biology into new definitions of health versus disease, we need to go deep into patients, not just doing the EHR analysis we should also do. And this need to come with major programs to profile patients longitudinally and at the highest possible resolution. Just look at how a patient that we are now tracking using those circulating single-cell RNA-seq analysis, how it is looking like over time with a certain clone that is detected here, because those techniques can also detect karyotypes. In 3 time points, look how initially it is almost undetectable, but it is detectable, and then the clone is expanding, or in another situation, a clone is being eliminated completely by treatment. So this is where we want to go to. That's the end. Thank all the people in the group, and again, Liran Shlush and his group for collaboration and funders. Thanks.